So starting with the polyadenylation. So what happens? We have a mature mRNA chain where we have the 5 prime cap and a 3 prime tail. So this 3 prime tail is called as the poly A tail, and nearly 250 adenosine residues are added in the tail. Now, this tail provides the additional stability to the mRNA. And how does this tail adds on to the mRNA chain? So there is a signal called as poly A signal that recruits both CPSF and CSTF, that is attached to the phosphorylated CTD domain of the RNA polymerase 2. Now, this CPSF and CSTF uh, has separate binding site. So poly A signal that has a sequence of AAU AAA is being bound by the CPSF, and the CSTF has a binding site of the GU-rich element that is downstream of the poly A signal. Now these regions basically, like when this protein binds, they form a loop, that is they both interact with each other and forms a cleavage site, that is poly A site, where the polyadenylation also starts. Okay, so this thing happens, then the CSTF is released. First the cleavage happens, then CSTF released, then the polymerase adds on to the this particular RNA chain, and this polymerase is template independent, and it doesn't require any template to add the nucleotides, and then it uses ATP, and it incorporates AMP to the running mRNA chain. Now, this particular polymerase first synthesizes nearly 10 adenosine residues. Then it moves on to produce more adenosine residues after the binding of the polybinding proteins. Now, this thing happens, then around 200, nearly 250 adenosines residue are added to the tail, and then the CPSF and polymerase both are released, thereby binding to the binding proteins. Okay, so length of the poly A tail is proportional to the stability of the mRNA. So as much as long the poly A tail would be, that would be the RNA would be more stable. And what are the functions? So it basically helps in the export of the mRNA from the nucleus, and it increases the half-life or the stability of the mRNA, and it assists in translation initiation. Okay, so moving forward to the next uh, RNA processing, that is RNA editing. So what happens in RNA editing is that it we can change any nucleotide sequence in the transcript. So as the editing means changing of any of the nucleotides. Okay. So what are the main functions of RNA editing? So it acts as a defense mechanism against the viruses and transposons. Okay. So sometimes because human body is really complex, so if we have to defend against the different viruses and the internal genome changing elements, so then these RNA editing come into picture. So they basically neutralize any of these harmful agents, and this basically helps in editing of the transcript that has been mutated by the genome. So without changing the genome, we are changing the transcript to produce a normal protein. That's the main thing. Now there are two types of RNA editing. First is the insertion or deletion editing, and other is the substitution editing. So we'll be discussing first about the insertion or deletion editing, then followed by the substitution editing. So what is insertion or deletion editing? They generally occur in the mitochondrial mRNA, and uridine editing is more common in this case. So they basically insert, we generally insert, but deletion is not seen in very common. It's like insertion is a common editing form as compared to the deletion part, and it is catalyzed by a protein complex that also have a RNA component involved, that is a 20S editosome. So it has two components, the RNA part and the protein part, as seen there. So the RNA part is called as the gRNA or the guide RNA, as in case of the CRISPR it is involved, and the protein part. So guide RNA, this decides the target where the mRNA to be modified. Okay, so it guides the proteins like in CRISPR-Cas9. So the guide RNA basically targets the site where it needs to be inserted or it needs to be edited. So, and the proteins have the endonuclease ligase and uridyl transferase. So uridyl transferase is would be the main activity of the enzyme involved, because the uridine has to be entered into that particular mRNA. So here is an example given. So this will be the normal mRNA where this particular adenine is present, but here the uridine is not present in the mRNA. Okay, so here the guide RNA, and this is the 20th editosome. So what will happen? It will break the bond between C and G by endonuclease activity. Then the uridyl transferase will come and it will add the uridine residue in the mRNA chain, followed by the uh, when the bond will be broken. So now what will happen? The ligase activity will again seal this bond after the uridine, after adding the uridine. So this will give us the normal mRNA. That will yield the normal protein. Okay, so this was the main theme of the insertion or deletion editing. Now moving on to the substitution editing. So substitution is basically conversion of either of the base to any other. So we have two types of substitution. One is transition and another is transversion. So transition basically involves changing of purine to purine or pyrimidine to pyrimidine. So this is the main theme of the transition, and then transversion involves changing of a purine to a pyrimidine. Okay, either of the thing can happen. Now, most common is the thing that is deamination based. So the bases are deaminased so that it will be converted to a new base. Now, two types of substitution we have seen in different examples. Like we have C to U editing, okay, that is done by the cytidine deaminase, and A to I editing. This is inosine. So this is done by adenosine deaminase. These two editings are generally seen in the substitution editing. So there is an example of the apolipoprotein B. So what happens? It forms like different isoforms in liver and in intestine based upon their function. Okay, based upon their need. So what happens here? The cytidine deaminase come into picture, and here is uh, sequence CAA in the apolipoprotein B gene. When there is no editing in the liver, it is normal, so it forms a protein that is made in the liver. Now, when this CAA is changed to UAA via cytidine deaminase, that is substitution editing, so this is converted into a stop codon. Now, this stop codon forms a truncated protein that is involved in intestines only. So, it is involved in different function as compared to the protein that is made in liver. So, here this is how the same protein gene forms two different proteins in two different tissues using RNA editing. Okay, and these two proteins are normal, no mutation is there. Just due to the function change, we the body is producing different kind of protein. Okay, so this was the case with the substitution editing. Now, um, this comes to the end of the mRNA processing. Now we'll move on to two more processings, that is rRNA and tRNA processing. So, in case of rRNA processing, we'll first see about the bacterial rRNA, followed by the eukaryotic rRNA. Okay, so as in the case of prokaryotic rRNA, so in case of prokaryotes, as we know, sorry, the prokaryotes RNA polymerase produce all kinds of RNAs, being it mRNA, rRNA, tRNA. Okay, and in case of rRNA, we have three kinds of rRNA present in the prokaryotic system. That is 23S, 8, 16s, and 5s. Okay, so these three RNAs, along with the tRNA, are produced as a single rRNA precursor that is 30s in size. So this is a pre-rRNA transcript that is 30s in size. Okay, now here, uh, this is the pre-rRNA transcript. Now what happens further? These bases are modified in the form by adding methylation and pseudouridine or dihydrouridine to the bases. Okay, so this transcript as like is modified based upon the position, and this modifications protects the RNA from degradation, and it like activates further signals for processing. So these lines, as you can see, are basically the methylation and pseudouridine or dihydrouridine sites. Okay, now what are these particular sites? 1, 2, and 3. So these are basically the sites for um, different RNAs acting. Okay, so one is RNase P, one is RNase M, and the other one is, sorry, RNase F, RNase E. So these three RNAs are basically involved in the processing of rRNA. So they have different cleavage sites, and now after cleavage, these intermediates are formed. So for the 16s, we have certain extensions at the 5 prime and 3 prime end that are trimmed by the nucleus later. First, these are separated from the original 30s transcript. Then in the tRNA also we have additional this thing regions. Then we have 25s for the 23s, and then we have the 5s RNA. Okay, after the nucleus cleavage, it forms the mature 16s rRNA, then tRNA, 23s rRNA, and the 5s rRNA. Okay, so here the major thing that needs to be remembered is the original precursor, that is 30s, the enzymes involved, and the basic thing is RNA M is involved in trimming. So these trimmings are done by the RNase M, and these cleavage first thing is done by RNase E and RNase F. Okay, so this is the main scenario, and then we have the like final thing occurs like after the cleavage of different intermediates. So this is the final mature RNA that is formed by the intermediates. Okay, so this was the thing about the prokaryotic rRNA and tRNA processing. Now moving on to the eukaryotic rRNA processing. So here, uh, first thing is that the splicing of rRNA is mediated by group 1 and group 2 introns. So these are self-splicing reactions, and these are extracted out on itself and based upon the transesterification reactions that we have studied in the ribozyme wala lecture. So you can go back and take a look on the process and how it happens. So the splicing of rRNA I'm not discussing now. Then the major thing is we have in the eukaryotic system we have four kinds of rRNA: 18s, 25s, 5.8s, and 5s. Sorry, 28s. This is 28s. So these are the types of rRNAs that are present here. These are formed from the precursor, that is 45S rRNA. So this formation of this particular like processing of the rRNAs occurs in the nucleolus inside the nucleus. Okay, so there the formation of ribosome happens simultaneously. So what we have is we have a 45S precursor of the rRNA, and this is a protein that is 90S pre-ribosome. So this is a pre-ribosome component that is totally a 90S. So it will involve proteins also. So that is why it is a 90S precursor. Then what happens? The SNOs, a small nucleolar ribonucleoprotein enters, and they modify the different bases in the rRNA chain. So by methylation, pseudo uridine, and other modifications. So they modify the bases, and then this triggers the cleavage of different uh, rRNAs. So these modifications are basically done to identify different rRNA molecules so that they will be cleaved as such. Okay. Now first cleavage happens that leads to the formation of the precursor for 18S and precursor for the uh, 5.8 and 28S, because these two are like packaged into different ribosomal subunits. Okay, so this forms the precursor for 18S forms a pre 40S ribosome subunit, and this forms a pre 60S ribosomes uh, subunit. So it also involves the 5S that is polymerized by the RNA polymerase 3. So it has a relatively different form of processing. Now, what happens after formation of this pre 60S and pre 40S rRNAs? There is additional cleavages of the removal of the extra RNA component, and then these like fully composed subunits are transported via the nuclear core to the cytoplasm. So the 40S is like bound with the 18S subunit and the different proteins associated, and the 60S is transported as the 5.8S, 28S, and the 5S rRNA subunits with associated with different proteins. And remember one thing, that 60S is the largest protein subunit passed through the nuclear core. Okay, so this can be asked in some of the competitive examinations. So this is the thing, and how the eukaryotic rRNA is being exported and it's being processed by the from the 45S precursor. Now moving on to the tRNA processing of the tRNA. So what happens in the tRNA? Now tRNA is being synthesized as a long precursor by RNA polymerase 3. Okay, and this is the primary transcript for the tRNA. So it's a very long thing. It What are the things that are extra here? This is the 5 prime extension, this is the 3 prime extension, and this is the internal extension. Okay, so now what happens? This thing is cleaved, this thing is cleaved, and this thing is cleaved. Okay, so this is the cleavage wala part, and trimming of the 5 prime and 3 prime end. Then we also modified the some of the bases are also modified at this particular loops. That is why different loops are formed in case of the tRNA. Okay, so first what happens? The 5 prime end of the transcript is removed by the RNase P. Okay, so this is the 5 prime cleavage. Then the 3 prime cleavage has two cuts for the RNase G and another for the RNase E and F, like we studied in the rRNA case. So This is the 3' cleavage. Then what happens? Then we have the CCA addition. Okay, so first this cleavage happens, then simultaneously after this cleavage, we add this particular nucleotides. Okay, this is done by tRNA oligonucleotide transferase. Okay, so this addition of ACC is done, and then different bases are modified. So here first base is dihydrouridine, and various places methyl group is also added in the chain on the guanine, cytosine, and adenosine residues, and this is pseudouridine. Now this splicing happens for this particular anticodon loop. Okay, that recognizes the anticodon. Now this particular thing is removed by different splicing reaction, and this is the mature tRNA formed. Okay, so this loop is called the DHU loop because here the dihydrouridine is there. This is the T-C loop. Okay, because here thymine and pseudouridine are there. This is the anticodon loop that contains the anticodon recognizing the codon from the mRNA, and this is the amino acid attaching site. Okay, so this brings us with the mature tRNA and how these thing happens. Okay, so this was the processing for the tRNA. Now moving forward, we missed during the initiation. We missed one thing. So in RNA polymerase 2 promoters, what was seen is the transcription start site was basically this was the Tata box. So the transcription start site was nearly 20 to 30 base pairs away. Okay, so it was not clear that the transcription start site is basically starting from the same base in every transcript, or it's the different base. Okay, so in various permutation combination, the scientists designed different things, and they found out that in one case these were the G position that act as a transcription in his start set. If they delete these particular base pair, then the transcription started from this base. Further, if they deleted these bases, then the transcription started after that. For example, here. Okay. Then they deleted 5 base pairs and added more 3. Then the transcription started from here. Okay. So this region between these two is nearly 20 to 30 nucleotides. Okay, similar experiments were done, and as you can see, the promoter region is basically reduced, but the transcription is starting nearly here only. Okay, so this means that the transcription start site will be different in different transcripts. Okay, so this is the scenario for this thing. So how will we detect the transcription start site in the eukaryotes? So there is one method called as the S1 mapping of the 5' end of the transcripts. Okay. So what we will do is, this is a DNA. Suppose this is the transcription start site. Now we like from the genome sequence that is available in the database, what we'll see, we'll first figure out the like enzymes that are present here. Okay, so that can cut these parts. Now, as seen in this particular figure, this is the transcription start site. We have a BAMH1 site here. So first, for the determination why we need these particular restriction enzymes is that first we have to develop a probe that will bind to the mature transcript that have a 5' end, so that we'll be able to figure out what is the start site of the transcription. So here we have the BAMH1 site. So what we did was we cleaved this particular thing with BAMH1 so that a smaller fragment is formed, that is with the transcription start site. Now we have another fragment of the SAL1. Okay, so what we did again was we first label the 5' end using the alkaline phosphatase, then a polynucleotide kinase, so that the ends of this particular thing is labeled. Okay, so this labels the 5' end. Hmm. This labels the 5' end of the RNA. Oh, sorry, for the DNA. So what will happen if the 5' ends are labeled? Then we'll able to detect the probe. So this was the main reason. Now, because both the ends of the DNA will be labeled, so SAL1 will be used to cut cleave off one of the sites. Okay, then we'll purify this particular fragment and denature the DNA, so that will produce a single stranded probe. Now we'll hybridize this particular probe with the transcript that is being formed, so that this probe will have a bit of the sequence similarity, that is from the transcription start site to the end of this probe. So this will bind to this probe. Then we'll use S1 nuclease. So the property of S1 nuclease is that it cleaves only single stranded DNA. Okay, it will not cleave double stranded DNA or double stranded RNA. So this S1 nuclease will not cleave the double stranded DNA or double stranded RNA. It will only cleave the single stranded DNA. Okay, so now when the S1 nucleus will act, this extra length will be degraded, and then we'll denature this part so that the RNA is separated from the DNA, and we'll run on a electrophoresis gel followed by sequencing. So we'll come to know what will be the start site for the this particular RNA. Okay, so this was the first method through which we can use or detect the start site for the particular transcript. Now, the second thing is primer extension. So, primer extension is an easy method. So, what we have to do is this is the DNA followed by the transcription. This will be the mature RNA, and as we can use any of the sequence or the in between this particular region. So we know the mature RNA would have something in between the mature RNA. So we'll design a primer such that it will hybridize to the labeled primer in between, and like this, and we'll extend the primer using reverse transcriptase enzyme. This is like a conversion of RNA to cDNA. Okay, not really cDNA, but basically convert reverse transcription. So this will happen, and the transcript, the cDNA will be formed, and then what we'll do is this will only go till the region where the 5' end of the RNA is there. Okay, so this leads to the detection of this particular thing, and we'll denature this hybrid and the electrophoresis, and then based upon the ladder, we'll sequence this end, and we'll find out what will be the 5' end of this particular RNA. Okay, so this was a bit easy method as compared to the S1 nuclease mapping, but yes, this can also be used to map the 5' end. Now, similar thing can be used to map the 3' end also. Okay, so we can design a primer such that it will bind here. and it can extend to up to here, or we can design a primer that is here. And similarly we can apply the same thing with the S1 mapping also. Okay, so this leads to our end of the eukaryotic transcription and the RNA processing part followed in the eukaryotes. So now we'll be seeing some of the previous year questions asked in different competitive examination, basically CSIR net, related to the RNA processing and eukaryotic transcription. Okay, so the first question is, HeLa cell extract was used to study transcription of a gene X having 6 introns. So this is a gene X that have 6 introns. Intron 1, intron 2, intron 3, intron 4, intron 5, and intron 6. Okay, so RNA polymerase 2 complex containing all associated proteins are isolated from actively transcribing system and subjected to the proteome analysis. Okay. Results showed that the presence of both splicing and capping enzymes in the complex. So what they did was they basically isolated the extract from the HeLa cell that have the RNA polymerase plus transcription factors plus different enzymes associated with the CTD. Okay, now they show that the presence of both splicing and capping enzymes in the complex. When transcription elongation was inhibited by flavopyridol, polymerase complex contained only the capping enzymes. Okay, so when elongation was not there, so capping enzymes were present. But when in normal conditions, both capping and splicing enzymes were present. Okay, when phosphorylation of the CTD domain of phosphorylated 2 was inhibited by a kinase inhibitor, the complex contained neither splicing nor capping enzyme. So when phosphorylation was inhibited, so now no enzyme was present. Okay, so the following conclusions were made. So the first point is transcription of gene X is coupled to mRNA capping. That is fine. And as we know that capping is co-transcriptional. Okay. Then we have transcription elongation is coupled to splicing. Yes. So the second point, when elongation was stopped, then only the capping enzymes were present, but not the splicing enzyme. So this shows that elongation can be coupled to the splicing. So splicing is also co-transcriptional basically. Then phosphorylation of the CTD is required for the recruitment of both capping and splicing enzymes. Yes, because when phosphorylation was inhibited, then no enzyme was recruited, neither for the splicing nor for the capping. Both capping and splicing of mRNAs occur simultaneously. No, this cannot happen because during the elongation wala part, only the splicing happens. Okay, so these things are not being told in the question itself. So option S is false. Okay, so identify the correct set of conclusion. So P, Q, and R are correct. So correct option is option A because both capping and splicing do not occur simultaneously. Okay, so the correct answer is option A. Now moving on to the next question. The 3' end of most eukaryotic mRNAs is defined by the addition of the poly-A tail. Okay, so as I told you, the poly-A tail is added to the mRNA. So this is the cap and this is the poly-A tail. Okay, processing reaction called polyadenylation. The addition of poly-A tail is carried out by the enzyme poly-A polymerase. Given below are few statements about this process. So poly-A polymerase is a template independent enzyme. This is correct. So we have to figure out what is the combination is true or false. So polymerase catalyze the addition of AMP from DATP to the 3' end of mRNA. No, this is not correct. Why this is not correct? Because here they are using deoxyadenosine triphosphate. Okay, so this is basically used for DNA synthesis, not RNA synthesis. So this option is wrong because DATP is not used in RNA synthesis. Now, polymerase is an RNA template dependent enzyme. No, this is also not correct. Polymerase catalyze the addition of ADP from ATP to the 3' end of the mRNA. No, because only the, like, this is the base, this is the sugar, and these are the three phosphate groups attached. Okay, this is the gamma, beta, alpha. So this portion is only incorporated in the growing nucleotide chain. These are released out. Okay, so this is AMP, and diphosphate is not there. So ADP will not come into picture anywhere. So this is also wrong. Then polymerase catalyze the addition of AMP from ATP to the 3' end of the mRNA. Yes, this is correct because AMP is added to this particular thing on the growing poly chain. Then polymerase catalyze the addition of AMP from DADP to the 3' end of the mRNA. No. Anything associated with RNA do not involve deoxynucleotides or nucleosides. Okay. So option 1 and option 5 is correct. So here the correct option would be option C. Okay. Moving on to the next question. Alpha amnatin inhibits only polymerase 1, only RNA polymerase 2, RNA polymerase 3, or all RNA polymerases. So, alpha amnatin basically inhibits first, like if we see the order, so first RNA polymerase 2, then RNA polymerase 3, then RNA polymerase 1. This is the order for sensitivity through alpha amnatin. Okay. So here the correct option would be option B and option C. And as you guys would have remembered that the RNA polymerase 2 is 1,000 times less sensitive as compared to RNA polymerase 2 for the alpha amnatide. Okay. But if this question is only single select, then RNA polymerase 2 will be the correct answer. Okay. So answer is B and C, but if only single option would be selected, then option B would be correct. Okay. Moving on to the next question. A promoter deletion study was done in order to determine the binding sites for a transcription factor on the promoter, which is activated on treatment with the drug X. The following constructs were made. So these were the constructs that are being made. One has the minus 1800 position. One has the mutated region till minus 1210 position. The last one has the second last one has the minus 868 position till there. The previous things are deleted, and the last one has the minus 432 position of the promoter. Now, what we are doing is we have attached this particular promoter to the luciferase gene, and drug X basically activates this particular promoter. Okay, so here we have seen that in case of the minus 1801, the maximum activity is observed, followed by in minus 1210, and the last two shows minimal activity. Okay, so following statements were made. Region between minus 1800 and minus 1210 contains the binding site for the activator. Yes, this can be seen because the minus 1800 one shows the maximum activity of luciferase when drug X was added. Okay, so yes, this option is correct because here the maximum activity can be seen. So this can be the site for the activator that increases the transcription rate by binding to the drug X, followed by the Q point. Region between minus 868 and minus 1210 contains a binding site for a repressor. So minus 868 and minus 1210 No, this can also be the binding site for the activator, because here the more activity of luciferase is seen as compared to the control, but less than the minus 1800 wala part. So here also there will be some activator site that have been knocked out in the previous region also. So this cannot be the binding site for the repressor. Okay, now we see for the minus 868 and minus 432 wala region. Here the activity is minimal in both the cases. So what can be deduced is, here the this regions can be the binding site for the repressor. Okay, so the region between minus 868 and minus 432 contains a binding site for the repressor. Yes, this can be the case because here the activity of luciferase is minimal. So we can deduce that a repressor has been bound to this particular region, so that it will not be able to activate the transcription, and it decreases the rate of the transcription basically. And the region between minus 1210 and minus 868 contains the binding site of the activator. No, because if this region would be there, if this region would be the binding site for the activator, then this graph would show the similar activity as compared to this. Okay, this can be the extended region for the binding of the activator, but this cannot be the fully activator. Okay, so the correct options would be P and R. Okay, so this region is for the activator and this region is for the repressor. So the correct option is option A. Okay, now moving on to the next question. In order to study the transcription factor TF2H, it was cloned from a large number of human subjects. So TF2H basically has a helicase activity, and also it helps in kinase activity also. Okay. Surprisingly, the subjects having mutation in TF2H also showed defects in their DNA repair system. Given below are the explanations. Um, DNA damage is always associated with transcription inhibition. No, this cannot be the case, because when transcription is happening, so that is not related to the DNA damage at all. So that can be inhibited by any other means as than the DNA damage. So this cannot be the case. Then TF2H has no role in DNA repair. According to the statement given, it when TF2H is mutated, then DNA repair system was also showing some defect. So this can also be not the case. In mammalian system, TF2H plays an active role in transcription coupled DNA repair process. Yes, this can be the case because, as the statement suggests, that TF2H mutation shows defect in their DNA repair system also. So R can also be the correct answer. Because of mutation in TF2H, transcription initiation is inhibited and incompletely synthesized mRNAs remain attached to the template DNA, leading to DNA damage. So this can be not be the case because TF2H the mRNAs are not synthesized first of all, because the initiation is already inhibited, and this does not lead to any DNA damage. Okay, this will recruit certain proteins that will cleave off the RNA and the polymerase and the different transcription factors, but this will not lead to DNA damage response inside the system. So here the correct answer would be R only. This would be the correct answer. Okay. Now, moving on to the next question. An eukaryotic cell undergoing mRNA synthesis and processing was incubated with P32 labeled ATP. Okay, so here all the three phosphates of the ATP are labeled with P32. Sorry, that is labeled with the beta position. So, as I have previously told, also this is the base. Then this is attached to the sugar. This is the first phosphate, second phosphate, third phosphate. This is the gamma, beta, and alpha. So this phosphate is labeled P32. Okay. Now, uh, where do you think the radioactive isotope will appear in the mature mRNA? Okay, so first option is P32 will not appear in the mature mRNA under any circumstances because beta and gamma phosphates are released during transcription. Okay, so this thing is factually correct, but here one thing is missing because in the first position the of the growing chain. So this is the first. For example, this is the first nucleotide, second nucleotide, third, and so on and so forth. So here all the three phosphates are attached and followed by the different nucleotides. So here no beta phosphate will be present in either of the growing chain, but here the beta phosphate will be present. Okay, so the first option is wrong. Then we have phosphate groups of the phosphodiester backbone of the mRNA will be uniformly labeled as only alpha phosphates are released during the transcription. No, because the beta gamma phosphates are released during the transcription process, only the alpha is incorporated into the mRNA chain. Okay, so this is not the case. The second option is also wrong. The third option, P32 will appear at the 5' end of the mRNA if only it has A as the first nucleotide. Yes, because we have only P32 labeled at the beta position of the ATP. Okay, so when capping occurs, this gamma phosphate is removed, and here the GTP having the alpha phosphate is attached to this particular thing. So here the beta phosphate remains. So if the first nucleotide is ATP, then adenosine, then only the beta phosphate will be present there and it will show radioactivity. Okay, so this is the case with the so this option can be correct. Now the fourth option, no P32 will appear in the mature mRNA because the 5' terminal phosphate of an A residue will be further removed during the capping process. No, because the gamma phosphate is removed, but the labeled part is the P32 process. Okay, so the fourth option is also wrong. So the correct option is option C. Okay, moving on. Eukaryotic mRNAs have an enzymatic appended cap structure consisting of a 7-methylguanosine residue joined to the initial 5' nucleotide of the transcript. Given below are a few statements regarding capping. Capping protects the mRNA from degradation by the 5' exoribonucleases, correct? So, this is the mRNA chain, this is the cap. So, here the 5' exoribonuclease cannot attract this particular thing, and here it is inhibited, so it is protected from the action of the 5' exoribonuclease. Then during capping the alpha phosphate is released from the 5' end of the nascent mRNA. No, the gamma phosphate is released. Okay, so this was the first nucleotide. It has three phosphates. This is the alpha, this is the beta, and this is the gamma. So this is released. Okay, so this is the wrong statement. Then phosphorylation mediated conformational change in the carboxy terminal domain of RNA polymerase 2 enables its binding with the capping enzyme. Yes. So what happens when capping needs to be done? The carboxy terminal chain of the RNA polymerase 2 is phosphorylated. Thus, it recruits all the capping enzymes, that is guanylyl transferase and all those proteins involved in the capping. So, this leads to conformational change in the RNA polymerase too. So, this option is correct. During capping, a 5' triphosphate bond is formed between the beta phosphate of the nascent mRNA and the alpha phosphate of the GTP. Yes, because this phosphate is removed and then the G alpha is added to the beta and alpha chain. So this forms the 5' trinucleotide phosphate group. Okay, so this is also correct. So the only incorrect option is option Q. So which of the statements is or are incorrect? So option Q is incorrect. So the correct answer would be option B. So this was the last question for today, and In the next class, we'll be discussing about the transcriptional regulation in eukaryotes, the mRNA decay, followed by the post-transcriptional modifications in the eukaryotic system. Okay, so next week might be the last week for the lectures. So stay tuned and thank you very much for attending the classes.